Hello students, welcome to Brilliant Classes. Today we are going to discuss NEET 2020 Biology question paper. Look at the first question. In light reaction, plastoquinone facilitate the transfer of electrons from. This question from 11th grade, chapter called what? Photosynthesis in higher plants, right? Look at the question properly. In light reaction, photosynthesis process can be classified into two phases, right? One is called light phase and one is called dark phase. Light phase or light reactions are also known as photophosphorylation. Yes or no, students? So this question from photophosphorylation. What is the meaning of photophosphorylation? In presence of light, ATP synthesis. So where it is takes place, as we know, it is takes place in thylakoid. Yes or no? So look at here. In light reaction, plastoquinone. What is this? What is this plastoquinone? If you able to recall your normal theory classes, PS2, non-cyclic photophosphorylation. Non-cyclic photophosphorylation starts with PS2. Yes or no? Look at here. This is chlorophyll A, reaction center, and accessory pigments are here, right? Total accessory pigments are present here, surround around the chlorophyll A. This is PS2, photosystem 2, right? Because this is a non-cyclic photophosphorylation, right? So it is PS, it starts with PS2 and it absorbs the wavelength is what? 680 nm. Yes or no, student? So when light is fall on these accessory pigments, they finally entered into, the light is finally given to where? Reaction center chlorophyll A. Now from this chlorophyll A, a pair of electrons, yes or no? A pair of electrons are excited and received by primary electron acceptor, PEA. Wait a moment. In light reaction, plastoquinone facilitate transfer of electrons from. So come on, tell me here. From PEA, it is entered into the short electron transport chain, right? So what is the first protein which take or which receive the electrons from PEA? Plastoquinone. Yes, so here it is plastoquinone. So electrons are coming from where? Primary electron acceptor to plastoquinone. Look at here the plastoquinone. So plastoquinone is transfer electrons from PS2 to a next protein in electron transport chain called what? Cytochrome B6F complex. Yes or no? From here, it entered into cytochrome B6F complex. Yes or no, student? So here, this is a second electron carrier. And moreover, one important point here, when electrons are carrying, when electrons are transporting from plastoquinone to cytochrome B6F complex, it will emit energy. With that energy is received by ADP. Yes or no? Here it is ADP, one inorganic phosphate, and it will synthesis what? Tell me, ATP. So this process is known as what? Photophosphorylation. Yes or no? So look at here. Option 1, PS1 to NADP. Option 2, PS1 to ATP synthesis, PS2 to cytochrome F complex. Option 4, cytochrome B6F complex to PS1. Plastoquinone is a carrier protein is seen in non-cyclic photophosphorylation only. So non-cyclic photophosphorylation starts with what? PS2. Yes or no? Look at here the PS2 to cytochrome B6 complex and cytochrome B6F complex to PS1. So come on, tell me here. Plastoquinone transfer electrons from, come on, from where to where? PS2 to cytochrome F complex. So our appropriate correct answer of this question is what? Answer 3, PS2 to cytochrome F complex. So from this cytochrome F complex, electrons are transferred to plastocyanin, right? Plastocyanin here, and from here plastocyanin, this pair of electrons are going to PS1. So here chlorophyll A, accessory pigments are surround around the chlorophyll A reaction center, and this is PS1. PS1 observe the wavelength 700 nm. Yes or no? Here 700 nm of light here, right? So this pair of electrons are observed by PS1 here. Again light fall on the accessory pigment. Finally light is observed by the chlorophyll A. Again a pair of electrons are excited and received by primary electron acceptor, and this pair of electrons are going to pyridoxine. Yes or no? FD, and from FD it eventually NADP plus. So PS1 and PS2 both are involving in non-cyclic photophosphorylation. Whenever you see the plastoquinone protein in the question, you would understand what this is: non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, electrons are carried from where to where? Electrons are transferred from where to where? Tell me. From PS2, photosystem 2 to cytochrome F complex. PS1 to NADP. PS1, look at here, the PS1 to NADP. Where is the PS1 to NADP? FAD is there, and plastoquinone is not there. So option 1 is not correct. Option 2 also PS1 it is starting. Non-photophosphorylation, non-cyclic photophosphorylation, it will not start with the PS1. So option 2 also ignore. Option 3, PS2. Look at here. It is start from PS2 and plastoquinone is transferring electrons from PS2 to where? Cytochrome F complex. Right? So the correct answer of this question is what? Answer 3. Look at the next question. Next question. The sequence that controls copy number of linked DNA. This question from the chapter called Biotechnology, 12th grade, Biotechnology chapter. Look at here what they're asking. The sequence that controls the copy number of linked DNA in the vector is termed option 1. Palindromic sequence. How many of you remember the palindromic sequence? Palindromic sequence in DNA means what? If you read from 5th prime to 3rd prime and 3rd prime to 5th prime, the nitrogenous bases are same, right? Look at here. Let me write the palindromic sequence. This is GAATTC. So what is the complementary strand here? CTTAAG. So which strand is this one? This from 5th prime to 3rd prime and 3rd prime to 5th prime. GAATTC. So this is called what? Palindromic sequence. And palindromic sequence is recognition site. At the specific point of palindromic sequence between guanine to adenine, how many of you remember? Exactly this place, EcoR1. EcoR1 act and they cut the DNA into 2 fragments, sticky ends. Do you remember, students? So palindromic sequence is one of what? It is a recognition site. So they are asking which sequence? They are asking copy number of linked DNA in our vector. So palindromic sequence is not a correct answer. So ignore this one. Recognition site. Recognition site means what? Restriction endonuclease enzyme working place in a specific point of a specific site of DNA, right? Like a palindromic. So it is not a correct one. Third, selectable marker. How many of you remember if I take extra chromosomal DNA in bacteria called plasmid? So one place is known as what? ORI. This is ORI site. Origin of replication, where exactly the replication start. Yes or no? Next, selectable markers. Selectable markers are selectable markers. Selectable marker antibiotic resistant gene, right? Tetracycline, yes or no? Or beta galactosidase enzyme we use as a selectable marker. But what they're asking in the question, linked DNA copy direct enserity line disease. So when we insert our desirable DNA into the plasmid, that DNA is going to be replicated into the number of copies from the particular site of a plasmid called what? ORI site. So ORI site is the replication site. Whatever the gene or whatever the fragment we are inserting into the vector, that DNA is going to become a copy from the particular region. That region is called ORI site. So number of copies of linked DNA is going to be increasing in a medium from the particular site of plasmid called what? ORI site. So the correct answer of this question is what? ORI site. Second question, right answer is what? Answer is 4 is the correct answer. Look at the question number 3. Question number 3. Look at the third question. The specific palindromic sequence which is recognized by EcoR1. Just now only I told you students in a previous question, right? What is palindromic sequence? Palindromic sequence means if you read from the 5th prime to 3rd prime and 3rd prime to 5th prime is same. But look at the options. If you look at the option, all are palindromic sequence you are observing. Look at here. Let me explain option number 1. In option number 1, what they are asking, look at here. 5th prime to 3rd prime, CTTAAG, right? CTTAAG. It is a palindromic sequence. 5th prime to 3rd prime and 3rd prime to 5th prime is same. But is this the site where the exactly EcoR1 work? Is this the site exactly where the EcoR1 cleave the DNA? I told you EcoR1 form sticky ends. EcoR1 restriction endonuclease enzyme cleave the DNA at a specific position. Where is the specific position? Between G to A. Look at here. If you take a DNA, this is the 5th prime and this is the 3rd prime. This is 3rd prime and this is the 5th prime. If you take CAATTC, exactly this place the EcoR1 work? No. G to A it will work. G to A it will cleave. So where is the G to A? Option A. Option 1, no. No G to A. Option 2, GG. It's not correct. Option 3, GA. You click it here. GAATTC is the first one strand of DNA, 5th prime to 3rd prime. Coding strand. Next. Template strand. Look at here. What is the complementary? C, T, T, A, G. Yes, we know this. Base pair rule. So, EcoR1 acts on exactly this place. So, it will form sticky ends. So, before you answer this question, be careful student. Because if you read the question, if you read the options, all are looking like a palindromic sequence. Look at here. This is a palindromic sequence GGAT, GGATCC, GGATCC. And look at the option GGAACC, GGTTC. This is not a palindromic sequence. Okay, fine. You can look at the 4th option is ignore. But not only you have to focus on the palindromic sequence, you need to also focus on which restriction enzyme, nuclease enzyme they are asking to work on. EcoR1 only cleave the DNA between guanine to adenine. So, this is the exactly EcoR1 work. EcoR1 work. So, sticky and it will form. So, the correct answer of this question is what? Answer is 3. 3 is the right answer of question number 3. Right? Shall we go to the next question? Question number 4. Look at the question number 4, what they are asking. This question also from the 12th grade. Human health and disease. Identify the wrong statement with reference. Wait a moment. I wanted to highlight which one they are asking in the question. Wrong statement with a reference to immunity they are asking, right? Wrong statement we need to identify. Option number 1. Active immunity. Active immunity is quick and gives full response. Wait. Quick, full response. What is active immunity, students? Active immunity, one type of what? Acquired immunity. When an individual exposed to the antigen in our body, antibodies will produce and they fight against with that antigen. Is this a slow process or is this a quick process? When any person is affected or any person is exposed to antigen, our antibodies naturally release from the B cells, right? And they fight against with the antigen. They neutralize. But this process will take time. It is the slowest process. It is not a quick process. But look at the option 1, what they given. Active immunity is a quick. I am thinking this option is not correct. Look at the second one. Fetus receives some antibody from the mother through placenta. If you are able to recall human reproductive system chapter, placenta transfer antibodies. What type of antibodies? IgG immunoglobulin. So IgG immunoglobulin can be transferred from where to where, tell me? From placenta to fetus. It is a right statement. This statement is correct. In case, if in this place, if they are asking, infant receives antibodies from mother, that is also correct. Through colostrum, mother milk, IgA immunoglobulins will go. Ready-made immunoglobulin they are. Right? Fine. So this statement is appropriate. It's correct. Option 3. When exposed to antigen, look at here, I already told you. When exposed to antigen, the antigen it could maybe in the form of living or it could maybe in the form of dead. Antibodies are produced in the host body. It's called active immunity. Before I start this question, I told you what is active immunity. When an individual exposed to the antigen in our body, antibodies automatically released from the B cell. It is called the active immunity. It is a very, very slowest process. Fine. So this statement also correct. Look at the option 4. When ready-made antibodies like IgG, IgA are directly given is called passive immunity. Whenever we are injecting direct ready-made antibodies into the individual, it is called passive immunity. It is right. This statement also correct. So which statement is wrong among these 4? Active immunity is not quick. It is what? It is a slow process. So this is wrong. So correct answer of this question is what? Option number 1 is the right answer about immunity. Shall we go on to the next question? Question number 5. Look at the question number 5. Experimental verification of chromosomal theory of inheritance. Wait a minute, students. They are asking what? Chromosomal theory of inheritance. I hope you remember this question. This is also from the 12th grade. Principles of inheritance and variation chapter, right? Chromosomal theory of inheritance first time experimentally was done by Morgan. Am I clear? T.H. Morgan, who called modern father of genetics. Option 1, Boveri, Sutton and Boveri. How many of you remember chromosomal theory of inheritance explained by Sutton and Boveri? But they are asking in the question experimentally on the Drosophila fruit fly. T.H. Morgan was a scientist, explained this theory. So the right answer is not Sutton and Boveri. The correct answer of this question is what? Morgan. So the correct answer of question number 5 is option 2 on Drosophila melanogaster fruit fly. Question number 6. Look at the question number 6. This question from the chapter called what? Mineral nutrition from the 11th grade. Look at what they are asking. Match the following concerning essential elements and their functions in plants. Essential elements and their function in plants. Option 1, iron, zinc, boron, manganese. The 4 elements are given, right? So look at the first one, iron. Iron is, as we know when we talk about the micro and macro minerals, Micro and macronutrients. Iron is essential for the synthesis of chlorophyll. Yes, so iron is essential for the chlorophyll synthesis. Necrosis. Do you remember when the chlorophyll is less in the plant, or when the chlorophyll is not properly synthesized, the disease is called necrosis. I hope you understand. Option B, zinc. What is the function of zinc? Micronutrient. Zinc is essential for the synthesis of one of plant growth regulator. What is that? Indole acetic acid oxins. So here zinc will go with what? Indole acetic acid. Option C, boron, one of the micronutrient. Boron is essential for what? Pollen germination on stigma. When pollen grains are dropped on the stigma, pollen germination is essential. Which element is essential? Boron. So boron is go with the pollen germination. Fourth option, manganese. Manganese helps in the light reaction, photolysis of water. So water is going to be break into hydrogen, electrons and also oxygen, right? Do you remember? Right. So manganese will go with what? Photolysis of water in thylakoidal membrane. So the correct answer of this question is 3, 4, 2, 1. Where is the 3, 4, 2, 1? 3, 4, 2 and 1. So the correct answer of this question is what? 1. Question number 6, answer is 1. Look at the question number 7, what they are asking. In a question number 7, this question is from the 12th grade. Again, biotechnology chapter. In gel electrophoresis, separated DNA fragments can be visualized with the help of. How many of you remember the term called illusion? Removal of or the separation of DNA fragments. So how can you take the DNA fragments until and unless you see or you visualize that DNA fragments, right? So in gel electrophoresis, separated DNA fragments can be visualized with the help of direct NCERT question. Acetocarmine in UV radiation, ethidium bromide in infrared radiation, acetocarmine in bright blue light, ethidium bromide in UV radiation. As we know, when we expose this gel electrophoresis plate to ethidium bromide in UV radiation, we can easily visualize what? Small fragments and large fragments of what? DNA. Right? So the right answer of this question is ethidium bromide in UV radiation. So question number 7 answer is what? 4 is the right answer. Shall we go to the next question? Question number 8. Look at the question number 8. Look at the 8th question. Name the enzyme that facilitates opening of DNA helix during transcription. How many of you remember this question from the chapter called what? Molecular basis of inheritance. One of the important chapter in 12th grade, right? So look at here. In the question, they are asking about transcription process, not DNA replication. If in the question they are asking about the DNA replication, which enzyme can open the helix of DNA? We would have go with what? DNA helicase enzyme, right? So if you look at that DNA, this is DNA. Look at here. So what is transcription process? Formation of mRNA from DNA known as transcription. So in this process, in transcription process, DNA helix is opened by the enzyme called what? RNA polymerase enzyme. Why and how RNA polymerase enzyme? If you are able to recall, RNA polymerase 2 can be synthesized mRNA in transcription. Yes or no? So RNA polymerase enzyme can initiate the transcription process in eukaryotes. So look at here, from here there will be an opening and the formation of single-stranded RNA called mRNA. So the formation of this RNA from the double-stranded DNA known as what? Transcription, right? Transcription process. So which enzyme is initiating the transcription here? RNA polymerase. During transcription, DNA polymerase enzyme, how many of you remember DNA polymerase enzyme can synthesize the new nucleotides during DNA replication? Yes or no? If you are able to recall, when the DNA is going to be opened by the DNA helicase enzyme in replication process, this is the 3rd prime, this is the 5th prime, this is the 5th prime, and this is the 3rd prime. Okazaki fragment. This is called Okazaki and discontinuous, and this is called leading strand, right? Continuous strand, lagging and leading strand, that we know. And formation of new nucleotides, formation of new, this is a primer, right? And after that, the formation of a new nucleotides, here also new nucleotide by the enzyme called, tell me, it is DNA polymerase enzyme. Yes or no? This formation of a new nucleotides by the DNA polymerase enzyme, but they are asking transcription. So option 1 is ignored. Option 2, RNA polymerase 2 can initiate the transcription process and it open the helix and a transcription process will begin. So the correct answer of this question is what? RNA polymerase. DNA ligase. How many of you remember DNA ligase can join the DNA fragments? Right? So the correct answer of this question is what? Answer is 2, RNA polymerase. Shall we go to the next question? Question number 9. Look at the question number 9. In which of the following techniques the embryos are transferred to assist those female, let me underline, those female who cannot conceive? What they are asking in the question, look at here. In which of the following techniques embryos are going to transfer? Embryo, look at here, embryo transfer those females who cannot conceive. We need to read a question properly. Question, option number 1, ICSI, intracytoplasmic sperm injection, right? This technique is when used. This technique is used when the male partner have a less number of sperms. Oligospermia, oligozoospermia, the condition, right? ZIFT, GIFT. This ZIFT is right? Fine, because what is ZIFT? From the donor, we are receiving one egg, right? This is the female gamete from the donor, right? This is from donor, female. We are taking this X, and from donor, come on, tell me, from male, taking sperms from donor male. So both are going to be fertile in vitro. Fertilization takes place in vitro. So in vitro, right? So what will form here? Zygote. So this zygote is going to be transferred into the female fallopian tubule. Look at here. This is the female reproductive tract and these are fimbriae, right? This is uterus, right? So here this will be transferred to where? Into the fallopian tubule. Zygote intrafallopian tubule transfer is correct. Fine. This is okay, but ICSI is not correct. That is in a male partner who having a less sperm count. Now, GIFT, gamete intrafallopian transfer, and ICSI. Look at the option number 3. ZIFT, zygote intrafallopian transfer, one method which is used, which technique is used in a female, the transfer of embryos into the female. Number 2, IUT, intrauterine transfer. So what is the difference between these two techniques? I'll tell you. If you take, this is the fallopian tubule and this is uterus, right students? So this is a uterus myometrium and the endometrium is highly folded like this, right? And glandular, this is the endometrium membrane, right? And this is cervix and vagina. Fine. So look at here. IUT means at the cells of, at the morula stage or at the 32 cell stage of embryo. In ZIFT, 8 cells to 16 cell stage of this zygote transferred to fallopian tubule. When the females are cannot conceive, those females are cannot conceive. Next, IUT means intrauterine transfer, which means they allow this zygote till 32 cell stage of morula. That morula will be transferred to where? Into the uterus. This is the difference between what? These two techniques. In one technique, they transfer the zygote into the fallopian tubule, and other technique, they transfer the embryo into the where? Uterine cavity. Am I clear? So, the female who cannot conceive, two techniques will be followed to transfer the embryos. One is ZIFT and one is IUT. So, the correct answer of this question is, answer is 3. Shall we go to the next question? Question number 10. Identify the basic amino acid. Simple question from the chapter called Biomolecules, 11th grade, right? Identify the basic amino acid. So how can we identify one amino acid is basic amino acid or acidic amino acid or neutral amino acid? If you are able to recall, the normal formula of amino acid, the general formula of amino acid is what? Carbon, 1 carboxyl group, right? And 1 amino group, NH2, and 1 hydrogen, and 1 alkyl halide, right? 1 R. Now, if the amino acid has more number of amino group than the carboxylic group, such amino acid is termed as a basic amino acid, right? So look at here, lysine, valine, tyrosine, glutamic acid. So look at the fourth option, glutamic acid. It's already given it is acid, so ignore this one. Glutamic acid is not correct. Tyrosine, aromatic amino acid. Valine, in valine, amino groups are equal to carboxylic groups. So this is a neutral amino acid. But if you look at the lysine, in the lysine amino acid, number of amino groups are high. Correct answer, the basic amino acid is what? Lysine is the correct answer. Look at the question number 11. Identify the wrong statement. Identify the wrong statement with reference to transport of oxygen. This question from 11th grade, breathing and exchanging of gases. So look at that, what the question is asking. Wrong statement we need to identify. Number 1, option number 1. Higher H plus concentration in alveoli favors the formation of oxyhemoglobin. Students, wait a moment. We need to understand at the alveoli how many factors are effect in the formation of a oxyhemoglobin. Look at here. This is alveoli. This is alveoli. In an alveoli, there are majorly 4 factors are involved, right? Indirectly, they're asking about the Bohr effect. Look at here. When air comes into the alveoli, in this air, partial pressure of oxygen is high. How many of you remember how much it is? 104 mmHg. Yes or no? It is 104 mmHg. Right? Next. Partial pressure of carbon dioxide in alveoli is 40 mmHg. It is less. It is less. Now, this is a blood vessel. So look at here. This is a blood vessel. So what happened here? Which blood vessel is go towards alveoli? Pulmonary artery. In pulmonary artery, oxygen content is less, carbon dioxide content is more. So in this one, what is this? This is pulmonary artery. In this blood, PCO2 is 45 mmHg. Yes or no? Next, PO2 is 40 mmHg. Diffusion takes place, exchanging of gases. Gases can be diffused from higher concentration, their higher concentration to their lower concentration. So now, come on, help me here. Now, oxygen is high, so oxygen can diffuse from alveoli to blood here, and carbon dioxide is diffused from pulmonary artery to alveoli. So 4 factors involved here. Number 1, partial pressure of oxygen, partial pressure of carbon dioxide, H plus ion concentration and temperature, lower temperature. Higher H plus ion concentration in alveoli. When higher H plus ion concentration, it is acidic. Yes or no? So when a higher H plus concentration, oxyhemoglobin dissociation takes place, not formation of oxyhemoglobin, it is dissociation takes place. Yes or no? In this place, exactly at the alveoli, now lesser high H plus ion concentration. This is a very important student. H plus ion concentration is less. So here, less H plus ion concentration at alveoli. So this statement is looking appropriate. It is a wrong statement. Fine. Look at this 2. Second option. Low PO2 in alveoli. Low PCO2 in alveoli. Look at here. Low PCO2, it is 40 mmHg only. Low PCO2, partial pressure of carbon dioxide in alveoli, favors the formation of oxyhemoglobin. Look at here. When low partial pressure of carbon dioxide, high partial pressure of oxygen can diffuse the oxygen into the blood. So oxyhemoglobin form. Right? More oxygen in alveoli, it diffusing into the blood. Right? And it is bind with the hemoglobin and oxyhemoglobin form. It is the right answer. So this statement is correct. Third option. Binding of oxygen with hemoglobin is related to partial pressure of oxygen. Yes, when partial pressure of oxygen is high, more binding of oxygen to the hemoglobin. Yes or no? Look at here. When the partial pressure of oxygen is high, more oxygen is diffused into the blood and it will bind with the hemoglobin. Right? So this statement also right. This statement also correct. Last, fourth option. Partial pressure of carbon dioxide can interfere with the oxygen binding with hemoglobin. Partial pressure of what? CO2, right? Look at here, the partial pressure of CO2. Partial pressure of CO2 can be interfere with the oxygen binding. So this statement also correct. So what is the wrong statement in this question, student? The wrong statement is H plus ion concentration in alveoli favors. So the correct answer of this question is 1. Look at the question number 12. Question number 12. Floridian starch has structure similar to Floridian starch. Whenever you read the question, in your mind, something is running, right? Rhodophyceae algae. Why I am telling this Rhodophyceae algae? Because in Rhodophyceae, red algae, the storage food material is Floridian starch. Yes or no, students? This is direct NCERT point. Floridian starch structurally similar to Mannitol and algin. How many of you remember mannitol is a reserve food material in Phaeophyceae, brown algae. This is a complex carbohydrate. Laminarian and cellulose. Laminaria also reserve food material in Phaeophyceae. Cellulose. Starch and cellulose. Floridian starch, structure wise, it is similar to amylopectin and glycogen. So, Floridian starch is structurally similar to what? Amylopectin and glycogen. This is the reserve food material in Rhodophyceae algae. So, question number 13. Let me go to the question number 13. By which method was a new breed Hessadele? Strategies of food enhancement. Do you remember this chapter? Strategies of food enhancement in animal husbandry topic. Hessadele of sheep formed by using Beckenary eaves and merino rams. So two different breeds cross between two different breeds called what? Crossbreeding. Very simple. Look at the option 1. Crossbreeding. Inbreeding. What is meant by inbreeding? Inbreeding means cross between two superior male and female cattle in the same breed. It is called inbreed. Now, outcrossing. How many of you remember this outcrossing? Outcrossing also the crossing between the animals, male and female, superior male and female, in a same breed, but they do not have a common ancestor in the 4 to 6 generations, right? And moreover, if you are able to recall, outcross is the method which can overcome inbreed depression. But what you are asking? Hessadele is a sheep breed in Punjab, first time. They cross between two different breeds and they produce one sheep breed called Hessadele. So we known as cross breeding. Why I am calling it a cross breeding? Because between two different breeds, crossing takes place. So such a breed is known as cross breeding. So what is this mutational breeding? How many of you remember mutational breeding? Yellow mosaic virus resistant plant. Yes or no? Powdery mildew. Mutational breeding plants. Right? So the correct one is cross breeding. Question number 14. Look at the question number 14. Match the following columns 1 and 2. And select the correct one. They are giving the different type of endocrine glands. This question from the chemical coordination and integration, 11th grade. Right? Look at the first one, what they are asking. Pituitary gland, thyroid gland, adrenal gland, pancreatic gland, right? First, Graves' disease, diabetes mellitus, diabetes insipidus, Addison disease. So, how can we do this question? Pituitary gland. Among these 4 in column 2, which one is correct to the pituitary gland? How many of you remember pituitary gland, posterior lobe called what? Neurohypophysis. Pituitary gland structure, right? And the pituitary gland is having a 2 lobes, right? One is called anterior pituitary and posterior. This is known as adenohypophysis. Hypophysis, which is the anterior pituitary, and this is posterior pituitary, which is called what? Neurohypophysis. Yes or no, student? And from this neurohypophysis, 2 hormones. One is called ADH, antidiuretic hormone called vasopressin. And second one is known as what? Oxytocin hormone. And this ADH hormone acts on nephric tubule. Do you remember? When ADH hormone is less in the blood, the condition called what? Diabetes insipidus. No regulation of what? Urine. No regulation of, no controlling of water absorption. No regulating of water absorption from the nephric tubule, especially from DCT. Yes? So, pituitary gland is going with what? Diabetes insipidus. Thyroid gland. How many of you remember? Hyperthyroidism. When oversecretion of thyroxine hormone leads to Graves' disease. Hyperthyroidism. Right? Oversecretion. Next, adrenal gland. Adrenal gland cortex secrete one glucocorticoid hormone called cortisol. Do you remember this one? This from the adrenal gland. What is that hormone? Cortisol hormone. Can leads to Addison disease. Mineralocorticoid, which we call aldosterone. Glucocorticoid, which we call cortisol. And sex corticoids. From the cortex of adrenal gland, three different hormones can release, right? One is called mineralocorticoid, one is called glucocorticoid, and one is called sex corticoid. And from the medulla portion of adrenaline, epinephrine and non-epinephrine hormones can secrete. So this is Addison disease. Pancreas, which we call heterocrine gland. Beta cells produce the insulin. When the lack of insulin in the blood, glucose percentage increase, which we call hyperglycemia. Yes or no? So hyperglycemia can lead to the formation of what? Diabetes mellitus, sugar disease. So what is the answer? First A3, B1, C4, D2. So what is the correct answer here? 3, 1, 3, 1, 4, 2. 3, 1, 4 and 2. So the correct answer of this question is what? 1. Question number 14, answer is 1. Look at the question number 15. Question number 15. Very easiest question. Sexually transmitted disease we need to identify from the following. Select the option including all sexually transmitted diseases. Option number 1. AIDS, acquired immunodeficiency syndrome. Yes, it is one of the STD caused by the HIV virus. Fine, I agree with this one. AIDS. Malaria. Is this STD, sexually transmitted disease, malaria? No. This is one of the diseases from the protozoa called Plasmodium vivax, right? Female Anopheles mosquito bite. So this is not correct. So ignore this option. Filaria, Wuchereria branchiofti, nematodal worm, Culex mosquito. So ignore this one. This is not a STD. Fine. Option 2, cancer. Cancer is sexually transmitted diseases? No. So this is not a STD. AIDS is STD. Syphilis also STD. Syphilis and gonorrhea. We already learned in the reproductive health chapter, right? So option 2, ignore. Option 3, gonorrhea. Nigerian gonorrhea. Yes or no? Next, syphilis. And also what? Genital herpes. Genital herpes, HPV virus. Syphilis, gonorrhea. 3 are sexually transmitted disease. Gonorrhea, syphilis, bacterial disease. Troponema pallidum, syphilis. Neisseria gonorrhea, bacteria, gonorrhea. HPV virus, genital herpes. So these 3 are sexually transmitted diseases. Fine, I go with the 3. Look at the 4 also. Gonorrhea, yes, which can transfer this disease from mother to baby fetus also. Next, malaria. No, malaria is not a correct answer. So ignore this one. So the correct answer of 15th question is what? Answer is 3.